Welcome back, guys. I hope that this weekend's assignment didn't completely scare you off of AP. I'm making this video like 2 weeks ahead of time, so I honestly don't know what's gonna happen with that. Hopefully we handled it well, and if y'all really hit roadblocks, you messaged me. So today we're gonna finish our talk on macromolecules. We're going to be covering nucleic acids, which we know store our genetic information. And we're going to be covering fats, which, let's be honest, we all know what fat is, okay? But we call those lipids. And then I'm going to talk to you guys about the lab report that you're going to be doing later today. Okay, so nucleic acids. So these are polymers made of nucleotides. So the monomer is a nucleotide. So what they do is they store, transmit, and express hereditary information. So what this means is like the instructions or the recipes to make every protein in your body come from your nucleic acids. So these are your genes. Okay, we've heard that term before. So they come in two forms in most life that we know of, DNA and RNA, or those big nasty words, and here I'm gonna show off just a little bit. Deoxyribonucleic acid is DNA, and ribonucleic acid is RNA. So those are some big, ridiculous science words, and you probably are not prepared for this. Okay, so just deep breath, you're fine. Okay, so deoxyribo, what that means is the sugar that is part of the nucleotide is deoxyribose in that one. And then with RNA, the sugar is ribose. So that's how we name those. And we'll talk a bit about those differences in a second. Just letting you know, those differences are going to be on your unit exam, 100% chance. So listen up. Okay. So nucleotides can form polynucleotides, which is just a string of bases. And then nucleic acids are the completed bit because there's actually proteins and things on there too, because nothing's ever simple in AP. All right, so nucleotides have three parts. A nitrogenous base, or it has nitrogen, okay, a 5-carbon sugar, and we can name that a pentose because like a pentagon has 5 sides. So pentose is a 5-carbon sugar, which can be either, as we said, deoxyribose or ribose, and then one or more phosphate groups. So this detail right here, I skipped past it. I've never heard this before, and you're not going to be tested on it, so we're not really worried about that, okay? All right, so our nitrogen bases, there's 2 types, a pyrimidine and a purine, okay? So pyrimidines are made of 1 ring with 6 atoms, okay? That's how we're going to distinguish those. And if you did your base pair sort, you may have seen some of these molecules before, right? Thinking back to the olden days of like a week ago. So cytosine, thymine, and uracil are all considered pyrimidines. Okay, they're 1 ring. Remember that thymine and uracil are kind of interchangeable, just one is only in RNA and one's only in DNA, if you dimly recall this from middle school. So thymine is the DNA one. You use T when you do DNA. Uracil is only in RNA, so when you're doing RNA, you have to use U instead of T. That sounds really confusing right now. We will get to the whole, what we do with those letters in a later unit, so no panicking right this second. You can panic later though, you have my permission. So purines have 1 ring with 6 atoms bonded to a 5-atom one. So we have these double rings here for purines. So that's our adenine and our guanine. And they don't really get into it in the AP curriculum as much, but there was a man named Chargraph, I believe, that he figured out that the amount of A always equals the amount of T and the amount of C always equals the amount of G, because we always pair a pyrimidine with a purine. You see how they're shaped is they fit together better, right? So he figured out base pairing rules because if they always happen in the same concentrations as each other, then they have to be the matching letters in the code. It's actually kind of cool how I figured that out. All right, 5-carbon sugar is bonded to a base. Like we said, it's deoxyribose in DNA, and RNA, it's ribose. So they differ in their structure and their function. The one in DNA makes like a little bit more stable of a bond there, and that's why they do that. RNA, it is what it is. Okay, so you see that there's just an ever so slight difference. Ribose has like an oxygen, and deoxyribose doesn't have an oxygen. Can you see what they did there with the wording? So you're gonna find out in science that there's a lot of big scary words, but that's why we're kind of doing those prefixes and suffixes, is when you break them apart, it's something so stupid obvious, like you take the oxygen away and all of a sudden it's a deoxy, okay? There's a lot of junk like that. Okay, phosphate groups. So a phosphate group is added to the 5' end of the sugar, which is attached to the base to form a nucleotide. So what they're meaning by 5' is when I am counting carbons in a sugar, I start at that oxygen at the top and I go clockwise. So the prime thing, it's how we're numbering carbons. So I don't know if you can see my arrow or not. I haven't watched these videos through because I hate the sound of my voice. Okay, so like on the ribose, we have an oxygen at the top. So the carbon to the right, this little angle right here, that's 1', right? Or no, it's 2', and they count their way. So 5' is over here on the 5th angle of it. So that's what they're doing. That's what they mean. So 5' and 3' is going to be important to us later because that refers to how we read the DNA. So that is a thing. Okay, phosphate groups link adjacent nucleotides. There's something called phosphodiester linkage. I'm saying that slow because I know some of y'all are taking notes. So we know di means 2. So there's something with phosphates and there's 2 of them. So there is a directionality. They go 5' to 3'. You're going to see that again. So 5' to 3' is how we read these things. Be prepared. Okay, so the nitrogen bases stick out and we have the phosphate sugar bone right here. They're making these bonds back here. So here's the 5' phosphate end, and then we have a 3' hydroxyl end. So like all right, just remember 5' to 3', okay guys? Okay, the sequence of bases along the DNA or mRNA is unique for each gene. So that's how we make genes, is the sequence of those bases. So if you see the letters, those letters are referring to the bases because everything else is the same. So this is going to dictate the primary structure of a protein. So each set of 3 nucleotide bases codes for 1 amino acid. And if I change one nucleotide, like one letter got changed, I can change what amino acid gets put into the protein. And once I change an amino acid out of the protein, it's not going to fold the same way. So that also changes the 3-dimensional structure of a protein. So I want you to take a second and appreciate that. How is that connected to our anchor phenomenon? Because you're going to be seeing that again later. How is it connected, this DNA, to say sickle cell anemia, right? So that is the thought you're going to have in your head. You're going to be writing a CER on that later. Knowing that each set of 3, like each gene codes for 1 amino acid, which changes primary structure, which can change 3D structure. How can we connect that? I hope that makes sense. If it doesn't, email me. Okay. So DNA consists of two polynucleotides. All that means is that there are two strands of nucleic acids that get bonded together. DNA is a double helix. So you've probably heard that or seen that before. The strands are anti-parallel, which means that they're basically like mirror images. So we have like 5' to 3', and then it's flipped on the other side, okay? So they're held together by hydrogen bonds between bases. So in the middle of the ladder, that's held together by hydrogen bonds. So another fun thing to think about is knowing that hydrogen bonds are what hold the DNA together. Why on CSI do they use like chlorine to get rid of evidence? Okay, give you something to think about for a second. So we got hydrogen bonds, right? We know that chlorine is a negatively charged ion, right? You pour bleach on it, that negatively charged ion is going to try to attach to the positive parts of those hydrogen bonds, right? It's going to try to form up with those. So it's going to unzip the DNA and make it unreadable. So fun fact, that's why that works. Okay, so cytosine bonds to guanine, adenine bonds to thymine. Remember that was Chargaff's rule we talked about a second ago. Do you need a second to write that down? Sorry, I know some of you are listening in real time. I know you can pause. Old teacher habits, yeah. Okay, RNA is single-stranded. It varies in shape. There's base pairing within RNA too, because the RNA has to be able to attach to the DNA when it's made, and also different types of RNA have to bond together. So like tRNA and mRNA. Again, I'm kind of getting ahead of myself. That's going to be more like unit 3 or 4. So we're not worried about that right this second. Okay. So these are just some concepts you'll probably see them like on a test. So the main differences between DNA and RNA. So that's going to be DNA is double-stranded, RNA is single-stranded. DNA uses deoxyribose. RNA only uses ribose. So yeah, those are some things you need to know. You're given a fragment of genetic material with the following sequence, ACUGA. Is this DNA or RNA, and why? So how can we tell? Remember, we said that there were certain bases that were, one was only DNA, one was only RNA. We said that was thymine and uracil. So uracil is the RNA one. So this one would have to be RNA. Okay, and this next part, I'm going to skip through it because this is actually part of a question of the day. So you don't need that right now. Okay, lipids. This part's short. I'm trying to get through this because you do have a lab report that you need to do today. Okay, so they're not truly polymers with these. I don't move myself. There we go. So they're generally relatively small. Okay, they're often not considered a macromolecule. I know when we teach biology 1 or life science, we generally throw them in there just because they're super important, and when else are we going to talk about it, right? So they are nonpolar or hydrophobic. This is why like oil doesn't mix in water. So there are types of lipids. We have fats, which we are super familiar with, and as someone who loves to bake, I am aware of fat, okay? I am aware. Phospholipids, we're going to talk about what that means in a second. Steroids, this doesn't just mean like the bad for you steroids like you hear that athletes get in trouble for taking. Those are anabolic steroids, which they are a type of steroid. But also there's a lot of hormones in your body that are steroids. So like estrogen, cortisol, testosterone, progesterone, all of those hormones, they're steroids for the most part. But also like when you get like really bad sick and they give you like Celestone or something, that's a steroid, an anti-inflammatory, those are also made of lipids. So it's just a category of molecules here. So fats are usually composed of glycerol and fatty acids. So the glycerol is technically an alcohol, which is why it has that OL at the end, because of its hydroxyl groups, which, remember, those are the OHs. You may have seen that when you were doing your sorting activity. Okay, and fatty acids have long carbon chains with a carboxyl group at the end. I'm going to give you a pro tip. If you are listening, if you're taking notes, I want you to write this. I don't know that this is on this slide, but I want you to write this, okay? If you see a carboxyl group, that COOH, that is often how we are able to tell something is an acid, okay? And we see that not only with fatty acids, you're also going to see that COO group, that carboxyl group on amino acids as well. That's why they're also acids. So that carboxyl group is important. I hope you're writing it down. That's on your exam, I promise you. Okay, so 3 fatty acids can join to a glycerol via ester linkage between a carboxyl and a hydroxyl group. This is, you might have heard on a package of mono and diglycerides. Well, there's also triglycerides, and that just refers to how many tails it has, okay? So different ones have different things. So they can be saturated or unsaturated, and you've also seen that on a food label. So saturated fatty acids have no double bonds in the carbon chain, and they have more hydrogens. So it's like they're saturated with hydrogen. Since they have all single bonds in the tail, they're holding the maximum amount of hydrogen they possibly can. So what happens is when we go to make like Crisco or margarine, we can artificially make them saturated. So they basically fire a whole bunch of hydrogen atoms into a liquid oil until it gets thick, like you know how Crisco is thick, and they force it to become saturated. Didn't know if you knew that, but it's cool. So unsaturated fatty acids, they can have one or more double bonds. So you might have seen like polyunsaturated oils, like on a label or something. That means, poly means many, so that means there's definitely more than like 2 of those double bonds. We find those a lot in like fish, like your omega-3 fatty unsaturated fats, like all that. Yeah. Okay, phospholipid. I told you we were going to get to it. So it's a major component of cell membranes. You may have heard the phospholipid bilayer before. Bi means 2. So the tail is hydrophobic. It doesn't like water, and the head is hydrophilic. So it tends to arrange itself where the hydrophilic part is sticking outward, and then the hydrophobic part is sticking inward toward each other. So that, okay. I wonder if I can turn this and show you on the board real quick. Like, can you see it? Yeah, I think this will work. Okay, so I'm gonna draw it big, so hopefully we can see this. Oh, you can't really see it really well. Let me stop share real quick. There you go. Okay, guys, so yeah, you get to see my face big for a second. So with the phospholipids, so here is the hydrophilic. And this is hydrophobic. Oh, it's backwards, gross. Okay, so the words aren't gonna work. Okay, so what happens is we got the phosphate groups facing out and these little hydrophobic bits facing in. And so it makes this double layer. And even when it's not in cells, if you just put these loose into a beaker of water, they're actually going to make little droplets configured like this all on their own. So, fun fact. Let's see if it'll let me screen share again. Sorry, guys. Sorry. Okay, cool. We're back. All right. So let's talk about steroids real quick. So they're lipids that have 4 fused rings. Unique groups attached to the ring determine the type of steroid. So like we said, testosterone was 1. So these 4 ring groups, you see this, it's probably a steroid. I probably am not going to test you on this, but it's a thing to know. If you're going to go into any kind of medical field, you're going to hit this hard and heavy later when you get in college. Okay. So I'm just going to go ahead and click and make sure this is all put up. This is a nice, succinct little table for you. You can pause it and put this into your notes. You're going to be asked about this along with some of the stuff from the previous chapter on your unit test. So this is like our last big lesson before your test. Please email me if you've got any questions, okay? You are going to be watching a video on something called the McMush Lab. There'll probably be a couple of them because I can't seem to find a good one that does both showing you the prep and showing you the method. So we're going to do a combination of the two. So our hypothesis, remember that we don't do if-then statements in AP. Some teachers on videos may still be teaching it that way. Just do it the way I taught you when you do your hypothesis. We'll probably have one just like a Happy Meal will contain the lipids, starches, and simple sugars or whatever. Something to that nature. Write down their procedure, what they did. It's really kind of a pity we can't do this in person. I nearly made a student throw up when we did this last year. It was pretty great. They're going to puree a Happy Meal and then test it for the different macromolecules. So I hope you guys have fun with that. Again, let me know if you have any questions, and I'll see you guys in the next video. Take care.